pathogen. So we develop these recirculating systems for housing salmon. And then we have these other pathogen containment rooms for running experiments. And one of the features that we brought to this was these kind of individualized experimental designs where we can do high throughput, large experiments. So the first capacity was these individual tanks, these 0.8 liter tanks where we can look at things like shedding kinetics of fish. Over the course of an experiment, we can run about 600 of those at a time. And then these larger 6 liter tanks for looking at things like survival studies. We can run about 300 or so of those at a time. And so with this comes a lot of samples. We generate typically thousands of samples for a given experiment. So we've really ramped up sample processing. So we developed these robotic assays for extracting RNA and DNA, and then using molecular methods like quantitative PCR and digital PCR for quantifying pathogen loads. So this has really moved into this high throughput aquatic pathogen studies. Most of the research is focused on ecology and evolution of infectious diseases in aquatic systems. So some of the topics that we've looked at are drivers of virulence evolution, pathogen management in aquaculture, such as vaccination, selective breeding, and culling, co-infection, diagnostics of emerging diseases, and pollution impacts on infectious diseases. I've been fortunate to work with a lot of individuals over the years, ranging from high school students to postdocs. You'll hear from some of those today. Our primary system of study is infectious hematopoietic necrosis virus, or IHNV. IHNV is a negative sense single-stranded RNA virus in the family Rhabdoviridae. It's got this characteristic bullet shape. It's endemic in salmon fishes in the Pacific coast, ranging from California all the way up to Alaska. Causes an acute disease, which results in necrosis of the kidney and spleen and ultimately death of the fish. Here you can see some clinical signs. They get exophthalmia, dark coloration, lose their swimming ability. Waterborne transmission is the main route for transmitting this virus. So infected fish shed the virus into the water column. The naive fish will come in and they'll pick up the virus. This pathogen is spread worldwide through aquaculture and is estimated to result in over $1 billion in losses for aquaculture, fisheries, and conservation. So one of the nice features of this system is it's a great laboratory challenge model. We can grow the virus in culture, and this allows us to conduct these controlled experiments where we expose fish to a controlled dosage using a natural waterborne immersion exposure. We can then track mortality using these group tanks over the course of an infection, or we can look at shedding. So we isolate fish into individual tanks, and then we can take water samples and measure the kinetics of shedding over the course of the infection. So we take those water samples and then process them with qPCR. Of course, you can do a variety of other things, look at viral loads, immune expression in fish, and things like that. But we focused a lot on shedding over the years. That's been a main focus of my program at this individual fish level. So this is kind of what the data looks like from one of those experiments. We typically have about 20 fish per treatment group, and we'll monitor the kinetics of shedding over that infection. And you see this really acute process where shedding ramps up very quickly. It peaks out around day 2 or 3, and then it kind of tapers as the infection progresses. And that clearance rate is really what differs a lot between those individual fish. So I don't want to spend a lot of time on this, just to give you a sense of the variation. But some of the things that we've done with this is we've shown that virulence tends to be correlated with shedding. So if we look at two isolates of the virus here, HV and LV, HV is more virulent, it kills more fish than LV. It also has a higher shedding rate. And this is really driven by clearance again. So the fish are better able to clear that low virulence virus than the high virulent virus. So this means that more virulent genotypes typically have higher shedding rates, and this likely equates to a higher transmission rate. You're going to hear a little bit more about this from Melina later on. We've then used our data to parameterize mathematical models to see how this translates to a population level. And what we've shown is that in the absence of practices like culling, so just sort of a natural scenario, the high virulent viruses tend to win in a population, and the low virulent viruses are driven to extinction. If we layer in a practice like culling, we can select for low virulence. So we can kind of eliminate that high virulent virus from the population and allow low virulence to have this selection advantage. We've also looked at things like co-infection and how co-infection influences virulence as well as vaccine efficacy. So if we co-expose fish to IHNV and Flavobacterium sarcophyllum, we see that survival is reduced. So when you look down here in our unvaccinated groups, you see that a lot less survival in the mixed infection compared to our single infections. The vaccination helps to increase survival, but it doesn't bring it back to the level of a single infection. So we get diminished vaccine efficacy in these co-infection trials. The mechanism here is really an enhancement of viral replication and shedding. So just focusing on the virus here, again, you can see that in the mixed infection, the shedding of the virus by this red line is enhanced compared to the single line, this blue-green line. If you vaccinate, you bring shedding back down, but not to the level of a single infection. There's still this kind of prolonged shedding period. We've also looked at how microplastics influence disease in this system. So if we expose fish to the virus as well as microplastics and look at survival, we can see that certain types of plastics, particularly nylon, reduces the survival and increases mortality caused by the virus. And we see a nice dose response. As you increase the dose of the nylon, you get a larger effect or more mortality. We've explored other systems. So we've done a lot of work in American eels and looked at emerging diseases in American eels. One of my students, Aman Kohli, she showed that Vibrio vulnificus was a causative agent for red sore disease in American eels. And the prevalence of that disease and the pathogen was driven by temperature warming. So in warmer environments, we get more disease. So this might be sort of a climate driver of this in the system. Another graduate student, Zoimo Warshawski, looked at how this emerging pathogen, a nematode, Anguillocoides crassus, that infects their swim bladders, the prevalence of that in Chesapeake Bay is rising. There seems to be this age stratification where the older age classes of fish have reduced disease loads, and this indicated to us that there was probably mortality in the field because they don't clear the infection naturally. So in order for there to be that age stratification, there had to be some mortality. All right, so just to finish that up, I don't want to take too much time. We really use a combination of in vivo experiments, molecular tools, mathematical modeling, and field studies to address aquatic animal health. And I've had a lot of individuals that I've worked with over the years, just a few folks that have been in the lab recently or worked through us on big projects. Couldn't fit everyone here. There's some people in the audience I know today that we've collaborated with that probably didn't make it up here, but lots of industry partners, academic partners, government partners, and funding from a variety of agencies. So thanks for listening, and I'm happy to take any quick questions.
Thanks, Andrew. We got a minute or two for a question if anybody has one. Feel free to unmute yourself or go ahead and put it in the chat. Dave Groman says, have you used the IHNV system to select for resistant salmon stocks? That's a good question.
Yeah, that was what didn't make it into my slides today. Yeah, we've been working on a project with some industry collaborators that they've developed some fish lines that are resistant to the virus. And we've tried to look at how that influences shedding. So trying to move away from just like this idea of being resistant towards disease and also resistant towards infection and shedding. So looking at different trout families and how they vary in their resistance. So that work hasn't gotten out yet, but it is something that we're really interested in.
Excellent. If there's no more questions from the audience, Andrew, thank you so much. That was really great and informative and nice to see the overview. I recognize a lot of faces from that lab. You forget those things over time. So
thanks a lot.
Next up, we have Melina, if you want to share your screen.